Hi everyone. Today we're going to talk about the Calvin cycle, which is the second step in photosynthesis. So yesterday we talked about the light-dependent reaction and how light and water are needed to create the waste product of oxygen, but more importantly to create the ATP and NADPH that would be needed for the Calvin cycle. So let's focus in here on the Calvin cycle today. So here you can see that there's going to be our next reactant, which is carbon dioxide, which is going to enter the Calvin cycle. It also needs NADPH and ATP in order for this process to happen. And at the end, we're going to get sugar, not exactly glucose yet, but sugar as a product. And it's the product we care about. Please ignore this water here. It's not part of the chemical equation, but it is something produced in this reaction. So the light-independent reaction, you need to know its location. It's happening in the stroma of the chloroplast. So this area here would be the inside space, kind of like the matrix of the mitochondria. This is the stroma of the chloroplast. Also, it needs carbon dioxide, ATP, and NADPH for it to occur. This is going to be the things that are coming from the light-dependent reaction. All right, when you look at this picture, it might seem a little overwhelming at first. We're going to break it down into 3 steps, one by one. Let's get started. So first, when you look at this picture, you might think, how many stages are in the Calvin cycle? So what you should notice is there's 3. First, carbon fixation, then reduction, then the regeneration of RUBP. Also, you're going to need to be able to describe each of the stages' inputs and outputs. We'll do this going through each of the stages individually. So let's start with the first stage. The first stage is carbon fixation. What that means is it's taking inorganic carbon and fixing it into an organic form of carbon, which would be the sugar molecule. So what happens in carbon fixation is that CO2, which you can see here entering at the top of the diagram, is going to interact with a molecule called RUBP. There's an enzyme called Rubisco, which is going to combine them together. Rubisco is a very old enzyme that's been around for over 1 billion years based on what we currently know. And it's what's going to fix carbon dioxide into an organic form of carbon. So here you can see carbon dioxide combining with RUBP, and the enzyme that's going to do that is Rubisco. So carbon dioxide is added to RUBP, and that is a 5-carbon molecule, RUBP. So you can assume with carbon dioxide added to it, it's going to catalyze it into a 6-carbon molecule. And the enzyme that does this catalyzation is Rubisco. So this 6-carbon molecule will eventually break into 3-carbon molecules called 3PG. You can see it here in the picture. So 3PG, how did we get there? So if you think about it, if 6 carbon dioxides entered this cycle and they combined with 6 RUBP, which is a 5-carbon molecule, that's going to give us 6 6-carbon molecules. Not exactly glucose yet, but just a 6-carbon molecule. This will then be broken down into 3PG. How many will we have then? 12, if you break it into 3 carbon molecules. So the 6 carbon molecules all get cut in half, so now we have 12 3-carbon molecules called 3PG. The next step is called reduction, so right here in the diagram. So here is our 12 3PG, and we're going to enter and start the process. Reduction, remember, means to gain electrons and hydrogens. So this ATP that was made in the light-dependent reaction and the NADPH will all be used to help reduce the 3PG into eventually something called G3P, which is the important sugar molecule that we're trying to get. So let's review. The 3PG is reduced into G3P. So here you can see the 3PG, which will be reduced into G3P, which is called glyceraldehyde 3-phosphate. You can just know it by G3P. So every 6 carbon dioxides, so if you're looking up here, needs 12 ATP. We add that phosphate to 3PG. Also, it needs 12 NADPH to reduce the 3PG into G3P. This is going to make 12 G3P as a product. But as you notice here, only 2 are going to leave the cycle from the 12. This is important to remember. Only 2 are going to leave the cycle. Now, the next 10 G3P, the ones that are remaining in the cycle, are going to go to do the third step, which is called regeneration of RUBP. This is going to join to make 6 RUBP, which is needed in the cycle to do the cycle again. So we have to regenerate this RUBP. 6 ATP are needed in order to regenerate the RUBP. So we're going to add a phosphate. So when the ATP breaks and it releases the phosphate, those phosphates are going to change it from RUMP to RUBP. So let's recap. To get 2 G3P as an output, it's going to require 6 carbon dioxides from step 1, carbon fixation. It'll require 12 NADPH from step 2, the reduction phase. And it'll require 18 ATP if you count the 12 from step 2 and the 6 from step 3. Now, just because we said 6 carbon dioxide entered this cycle, it doesn't always have to be that way. We're just teaching you the numbers so you can see how it breaks down. But let's say that we only need 1 G3P. How would the math change then? We would then need just 3 carbon dioxides in the carbon fixation stage, step 1. We would need 6 NADPH in step 2, the reduction stage. And we would need 9 ATP, which would count the ATP in the reduction stage, step 2, and the regeneration of RUBP in step 3. You're going to watch this video on Edpuzzle after you're done with my lecture here in just one more slide. This video is going to help you understand the Calvin cycle a little more. And what I mean that you don't really have to remember the numbers, but you do have to understand that there's multiple cycles happening at one time. As you can see in this picture, it's showing you 6 different cycles happening at once. And they're going to all be creating G3P, which can later be combined to create other types of polysaccharides, also lipids, also proteins. So watch this video. It has a few questions that you'll have to answer as you watch to check for understanding, and it's due in class today. And our last slide here is, what is the fate of this G3P? Why doesn't the Calvin cycle end in just glucose? Okay. Well, the G3P is a precursor. It's a precursor to make glucose. If you combine 2 of them together, you will get a glucose. And then if you combine many G3P together, you can end up with sucrose, which would be adding 4 G3P together, because 2 glucose would be a sucrose. Also, if you have a chain of these glucose, which were made from G3P, you would get cellulose, which is going to make up the plant cell walls. Also, if the plant makes too much glucose, it can store it as starch for later for short-term energy storage. Now, these are all polysaccharides, but also G3P is the precursor for some amino acids like serine. Also, G3P is the glycerol 3-phosphate backbone of the fatty acids, and that fatty acids will connect to make lipids, for example, triglycerides or phospholipids. So it's a very important molecule that needs to be made. So it's not just for making sugar like glucose, it's also going to help make all of these other macromolecules like proteins and lipids that our body needs. So remember, G3P can lead to glucose, which will lead to a polysaccharide. It can lead to some amino acids, which are our proteins. And it also is a precursor for making lipids. I hope this was helpful. Please remember to do the 12 test your understanding questions on the photosynthesis stages PPT worksheet and submit them by Thursday. Now go watch the other Calvin cycle video, Nature's Smallest Factory.